Hi everyone, my name is Arzoo Patel. Today I'm going to talk about the amino acids in beta amyloid. Currently working on this project in Dr. Armstrong's lab along with Beth and Uma. Before we get started, a little bit of background about the amino acids, which were thought to be irrelevant in biological system, but recent studies and research has shown the presence and the importance of the amino acids. And to further this, we're going to talk about the amino acids in beta amyloid, which is the peptide that is found in Alzheimer's disease patients. Alzheimer's disease is characterized by the depositions of these beta amyloid aggregates, which can be used as a prognostic biological marker for diagnosis. So generally, the peptide that is found in the brain plaques is 42 amino acids in length, and it has 3 aspartic acid residues at position 1, 7, and 23, and then 2 serine residues at positions 8 and 26. Now, it is shown that different post-translation modifications such as radicals and isomerizations and racemization can enhance this aggregation. And previous studies have shown the existence of DNL amino isoaspartic acid residues. The only issue is that these studies use acid hydrolysis for sample preparation, which can inherently introduce racemizations into the peptides itself. While we were working on this project, what we noticed was a trend that D-aspartic acid residues have higher sensitivity compared to D-isoaspartic residues and L-isoaspartic residues. So to further explore this phenomena, we wanted to look at just the peptides with the mass spectra detection. So we injected these synthetic tryptic fragments of beta amyloid peptides. So the peptide is cut into 4 pieces after tryptic digests, groups A through D. We will not be talking about group D in this paper because group D does not contain any aspartic or serine residues. So groups A through C, as shown here, have these modifications. So the 3 different fragments A through C were evaluated at optimized conditions for the all L wild-type fragment for each group respectively. The 3 modes of mass spectra analysis were conducted. The first was single ion mode, single reaction monitoring, and product ion scan. So single ion monitoring is when one mass to charge ratio of the peptide is analyzed, which helps indicate the ionizability of the peptides, while single reaction monitoring takes one selected mass to charge fragment. It then selects and then it fragments it, and then it selects one of its fragments. This again gives insight into ionizability and also fragmentation patterns. While product ion scan fragments selects one mass over charge ratio, it fragments it, and then it observes all the ratio. It observes and scans over all the fragments, which helps to observe the ratios between the most intense fragments of the native all-L peptides and the same fragment for the epimers and or isomers. So right here for group A, this is at 10 microgram per mil for all group A through C. And this is the data for SIM and OCRM at 100 microgram per mil for groups A through C. So for group A, we can see that the D-aspartic acid residue modifications had the highest instrument response compared in SIM and SRM for both the 10 and 100 microgram per mil. This suggests that the D-aspartic acid residues is more ionizable compared to the all-L peptide, L-isopeptide, and D-isopeptide. Group B peptides have 2 different modifications, and it can be seen that the native all-L peptide has the highest response, while in SIM and OCRM. All of the peptides were less ionizable in both SIM and OCRM at both concentrations. For group C, there are several epimers and isomers that have higher instrumental response than the native all peptides. For most peptides at 100 microgram per mil, the relative ratio of the instrument response are approximately the same. And here in the product ion scans, you can see that the ionizability and fragmentation is different depending on the location and the type of modifications. With each group of peptides, all epimers or isomers had the most abundant product ion mass to charge ratio peptide and similar ions for the next most abundant species. However, we can see in the SIM and SRM data that the instrument responds differently, so the ionizability is really affecting the instrument and the peptides at this. All in all, what we're seeing in a quick overview is that the mass spectrometry detection does not respond equally to isomeric and epimeric peptides. And to conduct a thorough analysis, each peptide, each isomer or epimeric peptides should be optimized individually because optimized conditions for all L peptides do not assure optimization and best response of the instrument to each isomeric or epimeric peptide.